Okay, so this is the presentation about how eukaryotes arose. So we're talking about how first life evolved. Prokaryotes were the first cells to come about. So how did eukaryotic cells with all their compartments come about from prokaryotes? Dr. Lynn Margulis, who was a professor at BU, she came up with this idea of the endosymbiotic theory. So we're going to talk about what she said. Endosymbiotic theory of eukaryotic evolution. The endosymbiotic theory of eukaryotic evolution was proposed by Lynn Margulis in the 1960s. An overview is that she basically said that these prokaryotic cells engulfed other prokaryotic cells as if they were going to eat them, right? But it was helpful, so they kept them around. Instead of consuming them, instead of digesting them, the cell that was engulfed actually provided some advantage to that cell, right? So they were initially still separate cells, but they just reproduced in tandem. After many generations, the two cells became increasingly cell-reliant on one another, and they could not survive without each other. So therefore, and we've got some evidence I'm going to show you, that the ancestors of chloroplasts are actually photosynthetic prokaryotes, and the ancestors of mitochondria are actually aerobic, heterotrophic prokaryotes. Endosymbiosis in a nutshell. So it starts with an independent bacterium, two of them. One bacteria engulfs the other. One bacteria now lives inside the other. Both of them are benefiting. We'll see how in a second. They're passed on from generation to generation. Right at this point, they're reproducing separately, but eventually they become reliant on one another, and this inside cell can no longer exist as a cell on its own. Here's another picture of this, right, showing time. The ancestral prokaryote with its DNA and cytoplasm. Also, eukaryotes formed because of these infolding of plasma membranes that eventually separated from the outside of the membrane. So that's how we get the inner endomembrane system. So Golgi body, endoplasmic reticulum, nuclear envelope, nucleus, those sorts of things. At some point in time, the prokaryote or budding eukaryote engulfed an aerobic heterotrophic prokaryote, and that became the mitochondria. And at some point, that cell engulfed a photosynthetic prokaryote that became chloroplast. So we know the mitochondria came first because every cell, every eukaryote has a mitochondria, but not every cell has a chloroplast or a plastid. So that must have come afterwards. These were the exact words of Dr. Margulis in her papers, right? She said endosymbiosis is a topological arrangement indicating that protracted symbiotic association, so symbiosis means they are dependent on each other, they are living together and working together, generates an interdependent relationship in which the sum of the parts becomes a new whole, right? So each prokaryote was individual, and now it's a new whole, it's a new eukaryotic cell. As such, endosymbiosis generates evolutionary innovation, right, where metabolic cooperation confers survival advantage. She says it very well. So why would they both have an advantage? What's the advantage? So if the main cell is heterotrophic, meaning that it gets its energy from outside sources, it consumes food, it's a consumer, having an autotrophic inner cell would be advantageous. So that's where if it engulfed a photosynthetic organism, that would be advantageous, right? If the main cell is anaerobic, meaning that it cannot use oxygen to break down sugars and get energy, having an aerobic inner cell would be advantageous. So that's where having a mitochondria would come in handy, right? As oxygen accumulates in the atmosphere, oxygen is actually very deadly. So being able to harness that oxygen and use it would definitely be advantageous, right? Eventually, after many generations of the main and inner cells dividing separately, living separately, they become so interdependent on each other that removing them causes them both to die. Now, the inner cell, what's the advantage for the inner cell? The inner cell gets protection, right? Normally, it's out there in the world getting eaten, obviously, because it just got eaten by this one. And so being inside and cooperating actually gives it an advantage of safety. Here's the evidence we have to support endosymbiosis. You can see there's a lot, right? If you look at chloroplasts and mitochondria, they are the size of prokaryotic cells, right? About 1/10 the size of a eukaryotic cell. They have double membranes, so we think that that inner membrane is the original prokaryotic membrane, right? It actually has very many biochemical similarities to a bacterial membrane. Chloroplasts and mitochondria actually divide by binary fission, which is how prokaryotes divide. They contain DNA that does not have histones, which we know is true of bacterial DNA. Also, the DNA in mitochondria are double-stranded and circular, just like in bacteria, just like in prokaryotes. They have that circular DNA chromosome. And they contain tRNA and ribosomes that are similar to bacterial ribosomes and tRNA molecules as compared to eukaryotic. They have different shapes and structures, even in terms of susceptibility to some antibiotics. So some antibiotics attack bacterial ribosomes can also potentially harm mitochondrial and chloroplast ribosomes as well. Now in humans, those are kind of buried inside, so it's hard for the antibiotics to get at them, but they can affect them in a lab. A little bit more evidence here. So the small ribosomal subunit of cyanobacteria is very similar to the small ribosomal subunit to that of chloroplast. So we think that the ancestors of cyanobacteria are the ones that became chloroplasts, right? That makes sense because cyanobacteria are the only prokaryotes that we know that can split water to produce oxygen gas as waste, so they're the only ones that do some form of photosynthesis. This is really cool. So some proteins, like ATP synthase, are a team effort between ribosomes in the cytoplasm and ribosomes in the mitochondria. So what this means is that because ATP synthase is a complex of polypeptides, it's not just one protein, it's many proteins, many polypeptides. Some of those polypeptide strands are made in the cytoplasm of a eukaryotic cell, and some are made in the mitochondria. And the polypeptides have to come together, right? So ATP synthase is actually a coordination between two different genomes. The nuclear genome contains instructions to build part of the ATP synthase, and the mitochondrial genome contains instructions to build the rest of the ATP synthase, right, through two different sets of ribosomes. So here's a picture of that, right? So you can see ATP synthase contains all of these different components, right? It's got all these, it's like a barrel, and then it's got this barrel down here and another kind of arm off the side. It's got a lot of pieces to it. And some of these pieces are made in the mitochondria itself and some are made in the cytoplasm. So what we think is that some of the genes, so this originally was made in the mitochondria, and some of those genes got transferred to the chromosome of the main organism, and now you can't make the ATP synthase unless you've got both parts. Finally, there's this idea of secondary endosymbiosis. Primary endosymbiosis is what we've been talking about, where a main prokaryotic cell engulfs a cyanobacteria, for example, and which eventually becomes a chloroplast, right? Some plastids, which are in the chloroplast is in the plastid family, have more than two membranes. So we think that some, like brown algae, for example, some algae have plastids with more than two membranes. So we think that this eukaryote engulfed another eukaryote, which formed multiple membranes. So all plastids are from cyanobacteria, but not necessarily directly.